Hi, everyone. Welcome to our Tuesday seminars on the cerebellum. Today, we will hear from Philippe Isope. Philippe studied with Boris Barber, Daniel Popa, Clement Lina, where he has been focusing on the link between granule cells and Purkinje cells pretty much all his career. He was the first to demonstrate that about 85% of the granule cell synapses onto Purkinje cells are silent. Then he discovered in 2020 this really interesting observation about the gradients and the biophysical properties of granule cells and how those gradients led to something that resembled a Fourier transform of the inputs, timing inputs to the cerebellum, and how they became this transformation to the Purkinje cells. So I'm so happy to have Philippe with us today. Philippe, thank you so much. Look forward to your presentation.
Okay, thank you very much, Reza. And actually, thank you very much for organizing this very interesting series. It will be a wonderful data set for the future, actually. And thanks to everybody, actually, to try to listen to me. If you don't hear very well, just raise your hand. I will try to speak louder. So I will try today to tell you a little bit about what we are doing in the lab since a few years now. It's about cerebellar synaptic connectivity, and actually one of the big questions we have in the lab, and not only in our lab, of course, is how it can relate to internal models. And of course, before I go into the data, I just want to introduce you to the people that made this big and interesting discovery, I hope, and especially very talented PhDs and postdocs, Antoine Valera, Ludovic Speth, Francesca Binda, Anaïs Brangeret, Theo Rossi, Kevin Dorgans, and Theo Gagneux, and also very important people, I think, modeler, or people coming from math and physics that help a lot to do modeling on our data and help us to understand what we are doing actually in the lab. Just to start, before I go into the data, I will tell you a bit how I consider or how I would define what is an internal model. Let's take the example of the motor control during voluntary movement. Actually, if you do an action, you have to plan the action, of course, and the motor apparatus has to execute. But as we know, it's too fast to make sure actually sensory feedback can be fast enough to adjust the movement on the ongoing movement. So the cerebellum, as many people and many groups have demonstrated before, is there to do a very important job. It takes efferent copies of the motor control, of the motor signal, of the motor program itself, and sensory information from the body current state, from all the body actually. And with these two kinds of information can compute predictions. And this prediction can be used to adjust the next step of the motor action, or directly make corrections onto the motor apparatus. This is actually what we call feed-forward control. To do this, and the cerebellum actually has to, build on very important neuronal maps. And these neuronal maps actually have to make this calculation pretty well. To do this calculation, of course, because our body is moving all the time, because we are changing during the development, actually, our muscles are changing. These neuronal maps needs to be adjusted to make the perfect calculation all the time. So for this, we need to be, the cerebellum has to receive information to adjust neuronal maps to make the good calculation. This is done through, for example, the inferior leaf that can give a feedback signal or teaching signal to adjust a microcircuit in the cerebellar cortex, for example, to modify the calculation. Using this, actually, the cerebellum can give the right answer to our motor coordination, for example. Of course, and all these neuronal maps actually should be computed all the time. And this is what I would say, for me at least, is an internal model. Internal models for me are these neuronal maps that make calculation to adjust or to make prediction of what we are doing all the time. This is embedded actually in a very simple circuit. I won't explain too much about this, of course, but the cerebellar cortex and the cerebellum itself is an array of functional units that could be defined by input-output topography. Typically, actually, climbing fiber inputs have a topographical organization and then define cerebellar modules that are called sometimes, and for specific reason, microzones. In this microzone, actually, climbing fiber directly contact Purkinje cell, which is the sole output of the cerebellar cortex. And this define, and of course, many anatomists actually have developed and demonstrated that all this structuration, all this topography define olivo-corticonuclear microcomplexes. This is one step. And then this topography defined by the climbing fiber inputs is also superimposed on this topography. Superimposed are some specific markers of the cerebellar modules. These specific markers, which are sometimes called zebrins, are expressed in many cases in cerebellar Purkinje cells. These Purkinje cells define very specific stripes in the cerebellar cortex. Fortunately, actually, climbing fiber boundaries and microzones defined by the zebrin patterning match somehow. And recent data suggested actually each of these microzones defined by the zebrin patterning could be not completely redundant. What I mean is that in this specific microzone defined by specific markers, we may observe specific transduction pathway, typically PLC beta 3 or PLC beta 4, and which can define specific functional properties. So the picture we have is not only that we have an array of functional units. This functional unit may be specific depending on the area. So, I mean, what we think about the functional universal transfer made by the cerebellar cortex could be probably extended a little bit to more specific functional properties. The second major input of the cerebellar cortex is the Mossy fiber system. Mossy fiber carry information from the whole body, from the cerebral cortex, for example, to bring the motor program, the motor command, but also from the spinal cord. And then they end up on the granule cell layer that send a long axon that can cross many Purkinje cells and also interneurons in the molecular layer. Granule cells actually can coordinate many of these functional units in the system. What is important actually is to note that Mossy fibers are connect redundantly granule cells in the cerebellar cortex. They can send information to very different microzones in the same lobule. Moreover, granule cells also receive input from different Mossy fibers. So we can imagine now that at this input stage, I mean the granule cell layer, we can have a huge computation made in the cerebellar cortex that then can converge to the Purkinje cell layer. Then, of course, all this computation is also embedded into a very fast input system. Actually, cerebellum has to, handle very fast movement, very fast coordination, and Purkinje cells are spontaneously active, and they can follow, as you can see in this picture from the group of Paul Shaderton. When whiskers are moving, actually Purkinje cell can increase or decrease their firing rate depending on the orientation. And they can follow very quickly movement and control movement very quickly as well. So that means that the cerebellum compute a flow of information at very high frequency. Okay, so now if the cerebellum makes calculation based on specific algorithm, and if we want to address the question of internal models in microcircuit, what we should observe actually in our recordings or in our data is maybe stereotyped synaptic organization in microzones that control specific and stereotype motor behavior. And we will take an example, which is adaptation of locomotion. This is the base of the work actually I will show you just after. And alterations of locomotor condition should influence these neuronal maps if they are related to this storage of internal knowledge. And later on, actually, we'll also give you a little bit of information about how fast this information could be encoded, and if this is pre or post-synaptic. So this is the main question, actually, we have in the lab since many years, actually. So as I told you before, these functional microzones could be specific and could be related to functional organization. So we decided to target only one zone in the cerebellar cortex to make sure we study all the time, animal after animal, slice after slice. We study the same topographical organization and the same inputs. So, and this is interesting because we have in the cerebellum actually data that could demonstrate that in lobules 3, 4, for example, and this is data made by the group of Tom Rigrock, for example, that could demonstrate that typical microzones, P1 negative zone, P1 plus, P2 plus zones in lobule 4, 3 to 4, actually are directly connected to medial cerebellar nucleus or lateral vestibular nucleus and control specific muscles in the hinterland, for example. And the group of Izumi Sugiyama also demonstrated how Mossy fiber inputs from the spinal cord or the pontine nuclei target specific zones in the granule cell layer. So we choose to focus on this zone, thanks to a transgenic mice actually provided by Izumi, in which we have venous GFP expressed in aldola C compartment of the cerebellum. And this is a very interesting tool actually that help us a lot to position our recordings in brain slices under the microscope to make sure we patch always the same neurons. And actually, this is a picture of what you see under the microscope. You see here an aldola C venous brain slice actually under the microscope. And you see here that we can identify very well the P1 and the P2 plus, and then position our recording and try to patch Purkinje cell typically in this area here that is highly involved in locomotor adaptation. And we patched Purkinje cell and we try to understand how, at least at the beginning, how the granule cells inputs have been selected during the life of the animals. Slices are done in the adult, of course, after all the development. And we try to understand how granule cells of Purkinje cell synapses are organized. We use uncaging of glutamate using a pattern light illumination system that can allow uncaging on a very small area of the granule cell layer, typically 20 micrometer pixels of illumination. This is important because then we can map using this excitatory input and inhibitory inputs when we excite granule cells. This is what one example actually of our recording made by Antoine Valera. This is typically one Purkinje cell, one slice reconstructed, one Purkinje cell recorded, and this is a grid that we could get after stimulation of each of the square of granule cell layer. You see in red current elicited by uncaged of glutamate in this particular position of the grid. And what you see here quite easily is that sometimes you have connectivity, sometimes you have big area of silent granule cells. And we can draw using these, 2D maps, and we can also project these maps onto a single line, which is based on the position of the granule cells in the mediolateral axis. Here you have the position of the Purkinje cell, and you see here granule cells that are connected or not to this particular Purkinje cell. Obviously, what you can see here, for example, is that you have big areas that are totally silent in gray, and you have hotspots of position highly connected to the Purkinje cell. This is very usual to observe this in a brain slice exactly at that position when we record Purkinje cell here close to the midline. Interestingly, these maps, or these connectivity maps, as we can call them, are not totally static. If, for example, we position a pipette in the molecular layer, apply a very simple protocol that is known to elicit plasticity, 1 Hz for 5 minutes, for example. After the baseline, when we redo the map after the protocol, we can observe modification. We see sometimes absolutely no change, so same connectivity between granule cells and the given Purkinje, but we see sometimes potentiated area, and more interestingly, new area connected, as if synapses were awakened between granule cells and Purkinje cells. We also see sometimes depression in some spot. So we can modify the map quite easily. And actually, we address whether this new site could be really awakening. So to do that, we did map, choose area totally silent, and uncage glutamate. I mean, once every 3 seconds for a few minutes. And this is the result of the data. Here, each dot is each line of dot is a cell, and the square is the mean. So after the protocol of uncaging every 3 seconds, we can observe now that we have connectivity between the silent site and the Purkinje cell. So we concluded from this that probably we can awake or at least potentiate non-functional synapses between granule cells and Purkinje cells. It's hard to tell if they are totally awakened, but at least we switch a site from non-functional to functional and to the map. Of course, one question is to say, okay, these are silent synapses, but are they deaf or are they mute? Do we have postsynaptic receptors, or actually maybe they do not release vesicles? So to address this point, Theo Rossi actually in the lab designed a very clever experiment. He was telling us that maybe we could image directly glutamate release into brain slices. So to do this, actually, he expressed a glutamate sensor into the granule cells. Okay, it's we call that iGlu sniffer for a glutamate sensor, and he expressed this sensor into granule cells, and then image granule cell into brain slices using a two-photon imaging system. And what you could see, when you stimulate individual granule cells in brain slices under the two-photon, you can watch, of course, individual buttons on the parallel fiber, and under a 20 Hertz burst of stimulation, for example, you can watch, transient change in fluorescence due to glutamate binding onto the iGlu sniffer. You can see that on individual episodes, but it's more, of course, it's more easy to observe that after averaging. So what you can see here is release of glutamate at a single button on parallel fiber in a brain slice. And the question was, okay, if we have silent synapses, maybe we will observe many buttons in the slice that are absolutely silent with no release of glutamate. But on the contrary, what we observe and what Theo observed is that if we follow a single parallel fiber that release at least at one button, actually all the other buttons of the parallel fiber release glutamate. And on the overall, 93% of the button release glutamate upon stimulation. And actually, this is not that we miss some buttons, because when we calculate the interval between two buttons that we observe, they match actually what has been observed in anatomy. So our conclusion on that is that probably all buttons release glutamate, but maybe there is no postsynaptic receptor at the synapse. Okay, so now what we wanted to address was whether these connectivity maps are similar between Purkinje or between animals. Of course, if there are internal models, or if this is storage of information related to internal models, maybe we should see a kind of stereotypy into this map. So this is what we did with pairs of Purkinje cells recorded, and then connectivity maps are established on these two Purkinje cells. These two Purkinje cells are colocalized, they are very close together. When you draw the map and you just project the map on the 1D side, you can observe that they seem very similar. And when you just do the correlation between each point on the grid, you see that they are highly correlated. So Purkinje, and this is observed all the time. Purkinje cells close by together have very similar maps. But this is not the case, and this is what you can see in this correlation matrix. If you take a Purkinje, for example, close to P1 plus in this area here, and you correlate it, you correlate the grid with a Purkinje cell distant from this area, there is no correlation. And that means that Purkinje cells that are close by together have similar maps, but different maps from distant Purkinje cell. And it's not linear, as you can see here. This is groups that you can observe between the midline and the P2 plus microzone, for example, in this area of the cerebellum. So that means that depending on the position on the mediolateral axis, you have different category of maps for specific Purkinje cell groups. And here, for example, you see the general map that we observe for Purkinje cell recorded close to the midline. As you could see before, with hotspots on local granule cells, hotspots on P2 plus, for example, and silent site close to P1 negative. And this is always the same. And what you have here is a summary of 18 animals and 29 maps. So that's using this trick, actually, we can demonstrate that these maps seems to be conserved between animals, because the group that you have here and the group that you have here belong to almost 18 animals. Okay, so that demonstrates that connectivity maps between granule cells and Purkinje cells seems to be specific, at least from one specific area of the cerebellum, and very conserved between animals. What about actually the other cell types in this area of the cerebellum? So we recorded interneurons, molecular interneurons, and we also observed, conserved connectivity maps between granule cells and interneurons, but they are slightly different than the maps that were done with Purkinje cell. For Golgi cell, for example, you have also very different features. And actually, what is very simple with Golgi cell is that there is only local granule cells that are connected to a given set of Golgi cells. So, the grid and the connectivity maps are very different. So, on the overall, if you combine all together, what you have is that Purkinje cells have a certain conserved organization of connectivity maps, molecular interneurons also, but slightly different than the Purkinje cells, and Golgi cells, totally different connectivity map. Okay, so now what about real inhibition? Okay, so we could also record Purkinje cell in the same, using exactly the same paradigm, but also record the inhibitory input elicited by granule cell stimulation, by granule cell activation by glutamate, glutamate. Okay, so this is what you have here. This is a grid in which you have an area that elicit excitation in blue, or inhibition in orange, or both in green, or being silent. And this is the map that you end up with for excitation and inhibition. As I just showed before, they are conserved, but they are different, meaning that potentially granule cells can activate interneurons that inhibit Purkinje cell, but that do not excite directly Purkinje cell. So first conclusion of this work, actually, what we have demonstrated is that connectivity maps between granule cells and specific group of Purkinje cell are conserved. They can be modified. Synapses can be awakened. All synaptic buttons actually release glutamate. So it looks like, synaptic weights are more controlled by the postsynaptic side. And also inhibition and excitation can be dissociated. And actually this suggests us to make a prediction based on this. Maybe Mossy fibers can contact different groups of granule cells and activate differently the Purkinje cell through excitation and inhibition. And actually, granule cell temporal encoding could be also involved in the situation. So to access and to address this point, we express channel rhodopsin in precerebellar nuclei, in Mossy fibers. And on brain slices, we could then activate specifically individual Mossy fibers and record Purkinje cell. And what we observe, actually, strikingly, is that when you record excitation and inhibition in Purkinje cell, actually, sometimes you see what you expect to see. I mean, a sequence of excitation, then inhibition. But many times you can observe some kind of weird sequences, and even sometimes inhibition coming before excitation. So this demonstrates that in our system, when you elicit an action potential on a Mossy fiber, you may excite different groups of granule cells that are connected differently to the Purkinje cell, eliciting excitation, maybe one group eliciting excitation, another group eliciting essentially inhibition. And then the temporal organization of the EI sequence could be highly modified and highly specific in a given Purkinje cell. And in summary, here what you have is differences between excitation and inhibition latencies, whether you stimulate only granule cells, directly granule cells with electric stimulation, or if you stimulate Mossy fibers. And what we see is that the relative latencies between excitation and inhibition could be very different if you stimulate Mossy fibers. So that brings actually into question how temporal encoding and how information processing at the level of temporal code could be done actually in this system. Actually, why temporally decorrelated EI sequences could be important for Purkinje cell discharge, for example. And actually, we wanted to address this point a little bit. Why I'm saying that? Because you probably know, but because cerebellum and Mossy fibers speak at very high frequency, I mean, synapses at this high frequency mode adapt in a burst. Typically, this is called short-term plasticity, and this is quite a lot of data actually on the granule cell to Purkinje cell synapse or granule cell to molecular interneuron synapse, for example. Here you have an example of inhibition and excitation recorded when you stimulate individual granule cells. You see that excitation in a burst facilitate, and this is summarized in this example here, when you just do the ratio between the amplitude of the peak N and the first peak. On the other hand, inhibition tends to depress. Okay, so at each position in a burst in a train, EI balance in Purkinje cell is modified. So synaptic weights basically are modified depending on the position in a burst. So if you adjust and if you change the relative latencies, of course, you may affect a lot how granule cells will influence Purkinje cells. And moreover, we've demonstrated actually a while ago that this short-term plasticity, this is in this example, short-term plasticity recorded at the granule cell to molecular interneurons. Okay, so this is pairs of recording between a molecular interneuron and stimulation of a single parallel fiber. What we could observe is that the profile of discharge between individual parallel fiber and molecular interneurons could be very different from facilitating to non-facilitating, and you have a summary of the different classes that we observe in the molecular interneurons. Interestingly, this is not a continuum. We could observe specific classes actually when we could sort this profile. And this is demonstrated, for example, by the use of a specific knockout of a molecular determinant of a vesicular release. For example, we use synaptin 2 cow mice in which we knew that the profile of discharge of vesicular release could change. And this is the case in our data. But more importantly, when we use these knockout mice, we observe that a group of profile totally disappeared. So from the 4 classes, we end up with only 3 classes. So this demonstrates that short-term plasticity is not random, it's very regulated, and it's linked to molecular determinants in the cerebellar cortex. And to end up on this temporal, processing information, I will end up to this with this picture. This is how this short-term plasticity can affect Purkinje cell discharge. Okay, this is very a basic experiment. We recorded Purkinje cell in juxtacellular, then we could patch them to get the synaptic input, and we stimulated granule cells. And in many cases, what we observed upon granule cell stimulation actually is, an increase in the firing rate. Of course, this is direct excitation of Purkinje cell. On the raster plot, you see that you have more spike. And if you do the net increase in the spiking behavior of the Purkinje, you have more spike. Fair enough, it's excitatory. But in 15% of the case, actually, we observe the contrary. Granule cells excitation that lead to Purkinje cell decrease in firing rate, probably because EI balance is more oriented to inhibition in the feed-forward inhibitory system. But in many cases, more than 40%, depending on the number of spike we do in the simple burst between 3 and 7, for example, 1, 3, 7, we could observe that synapses, meaning some granule cells that elicited initially more inhibition, as you can see on this net decrease in the firing rate, when you increase the number, when you add the number of more stimulation in the train, you switch them to an excitatory system. And this is all due to the change in EI balance due to the short-term plasticity. Just remind, excitation facilitate, inhibition depress. So if you, the duration of the burst totally control how will be the influence of granule cells to the Purkinje cell. So this has been also confirmed by a model done by Arvind Kumar on this publication. So to conclude on this small, actually, temporal information processing data, what I would like to say is that we should think about combining the spatial organization of the inputs in granule cell layer, also to short-term plasticity monitoring to understand how information will be processed in the cerebellar cortex. And actually, I just want to remind you the data by David Di Gregorio showing that Mossy fiber short-term plasticity influence how granule cells discharge during a burst of input. Okay, just to finish and on this presentation for the next 5 minutes, I would like to jump back to the initial question. I just showed you before that we have connectivity maps that are conserved between animals, but I didn't show you whether these maps are linked to a specific context, meaning that do they provide information for a specific adaptation of locomotion? Okay, so this is what I will try to show you now by some data in which we altered locomotion or we train mice into a specific paradigm. So we used 3 specific conditions. We impaired locomotion using a cuff on the synaptic nerve on the right hind limb, which make the mice lumping for a few days before after the surgery, before they readapt. Okay, they keep the cuff, but they readapt after 1 or 2 weeks, and they can walk perfectly well. On the other side, we train some mice in a wheel for 3 weeks before we do the experiment. So this is completely opposite way to alter locomotion. Impairment, or what we could say, improvement, or at least modification of the type of locomotion the mice can do in that cage. Okay, and the sham actually is just a surgery that we did for the cuff, but without positioning the cuff. Of course, when you put mice in a wheel, they like to run, and actually they improve day after day. So this is after 3 weeks, they can do about some mice actually could do 1 kilometer per hour in the wheel. On this group of mice, actually, we did connectivity maps at 2 time points, after a week or at the end after 3 weeks. Okay. On the other side, we monitored actually the balance of the mice that were cuffed, so the one that had impairment in locomotion. We just monitored the balance on force pressure sensor on a corridor. So what you see here is that this is the alternating balance between left and right when the mice are walking on the corridor. Okay, so in normal condition, of course, they alternate between left and right, put the weight on one side and on the other side. When they are cuffed, actually, they actually put all the weight on the left part because they are cuffed on the right side. But after 3 weeks, they totally recover. You don't see anything on the force pressure sensor. They can work perfectly even with the cuff. Okay, and if you summarize actually the change in balance during these 3 weeks or even a month, what you have here is just the imbalance on the left or unbalance on the right. The red curve is how cuff mice actually adapt to the situation. The first week, actually, they are unbalanced on the right, and the second week, they are unbalanced, they compensate on the left before recovering completely their balance. What is interesting here is that all the group, the 10 mice, basically readapt in the same way. I mean, the same kind of unbalance actually was observed in the group of mice. Again, we did connectivity maps after the first week and after total readaptation of the locomotion. Okay, so what we observed, to make it short, is interesting things. So what you see here is a grid of connectivity map recorded in a Purkinje cell located always at the same position, so close to the midline in lobule 3-4. What we observe, you don't need to dig too much into the picture, but just watch that in each condition, train, sham, and cuff, early and late. Early means after a week, late means after 3 weeks. The organization of the connectivity map has changed. But again, they are very similar between animals, because this is a summary of 10 animals, at least, in each of the conditions. So in a given condition, the map has reorganized, but in a similar way. And between conditions, maps are totally different. For example, in some condition, actually, you don't see any more input from the P2+, and you have an appearance of an input in the P2 negative bands, typically. So what is interesting here is that this is conserved, but different between conditions. Okay, and if you just summarize, and if you just sum up all the synaptic inputs in these maps, what we observe is that for invasive conditions, such as cuffed and sham, because they got the surgery, the overall total synaptic charge observed in the map increased after all. Okay, so this is, I mean, what we show here, that the connectivity maps are then conserved between the locomotor condition. But if you see, I mean, more precisely, you could observe, I mean, what you see in light gray here is a median absolute deviation from the histogram. So there is relatively high variability still between animals, although conditions are a very strong argument for a connectivity map organization. There is still variability between individuals. So we asked actually how we could take advantage of this individual variability and try to see if we could correlate individual maps with maybe a condition or a specific feature observed in a condition. So for this, we end up with this option of transforming graph in our connectivity map into a graph network. So we tried it very simple, and thanks to Damien Battaglia and Jyotika Baruna, so we developed this very specific analysis. We took connectivity maps from our recording, of course, and then we split these maps into columns of connectivity. X and X prime are columns of granule cell, Purkinje cell connectivity in granule cell layer. And we compare them all together. So each column is correlated to all the other columns of the connectivity map. Okay, so we end up with a matrix that could be reorganized, and we applied graph metrics to evaluate actually how these individual columns, which now are called nodes in the graph language, how these different nodes and these different groups of nodes are structured in the connectivity maps. Okay, so typically, are they organized in modules? Are they clustered? What kind of relation can we observe between nodes? Typically, is there a node highly connected to other node inside the module or outside the module? Okay, this is participation. Or we could observe whether nodes are highly associated to same type of nodes, and so on. And this different kind of metrics increase the dimension of the connectivity map, so we can try to correlate better with behavior. So this is what we did. This is the framework that we use. Behavioral observation, extracting features, building the map with the patch, okay, building the graph network, and try with machine learning to predict behavioral feature from individual maps. Okay, so we did that in two different situations. With a supervised learning algorithm like the random forest to see whether connectivity maps can predict the condition in which the map have been recorded. For example, cuff, sham, or control mice. And what we observe in the confusion matrix here is that using these graph metrics, as opposed to just synaptic weights, we could recover completely, and we could predict, given the connectivity map, what was the condition in which this connectivity map was recorded. Okay, as if there was in this connectivity map enough information to build a model to catch this information. Okay, and if we shuffle the connectivity actually into the maps, actually we get the total lost. I mean, we are lost totally in the accuracy of the modeling. Okay, and lastly, and this would be the last information, if we take this connectivity maps and transform that into graph metrics, so this is our inputs, okay, and use a linear model, basically a regression model, to try to predict a simple output. In that particular case, the balance of the animals in the cuff, for example, 2 weeks after the surgery. The predicted values correlate perfectly well with the real value observed during the life of the animals. And what you see here is basically all the animals. In red, animals that were cuffed, and in black, animals that were sham. And we have a very nice correlation between predicted and real data. So for us, these data are very strong to, let's say, to demonstrate that there is a clear link at the individual level, at the level of the animal, between how the synaptic organization, how synapses are organized in lobules 3, 4, in this particular area of the cerebellum, and the real behavior of the animal in a given condition. So, as general conclusion, maybe I would like you to keep in mind just this few information. Synaptic connectivity maps are stereotyped in a given area of the cerebellum, but they can adapt to locomotor conditions. Okay, microzones are coordinated by this granule cell input. Second, post and presynaptic mechanisms influence information processing. And one important mechanism is short-term plasticity that can enhance the dynamic range of influence for Purkinje cells. Third, graph analysis can also help to dig into the structuration of the connectivity map, okay, and give very insightful information to understand how the cerebellum encodes information. And lastly, I hope I could convince you a little bit that cerebellar cortex, actually, at least in this area, store one piece of internal models associated to locomotor adaptation. And this is, for us, I mean, a good demonstration that we are on a good way to understand how the cerebellum encodes locomotor adaptation. So just to finish, I want to introduce, the sponsors, the ANR, the FRM, and the Fondation de France, but more importantly, again, the people highly involved in this data. Antoine, Ludovic, Francesca, Anaïs, Theo, Kevin, and another Theo. And I would like to thank you for your attention.
Thank you so much, Philippe. Let's open it up with questions. Maybe I can start first with asking you the cuff experiment and how surprised were you regarding the results that you got? And I guess if you could link it back to the way the maps were measured and how the cuffing of the animal, you said that the animal behaviorally begins to rely more on the other side, the contralateral side. So what would be the prediction of how the maps should change given the behavioral results?
My suspicion or my expectation is that in this area, the midline actually is really involved into proximal control, proximal muscles control in the hinterland, for example. And it turned out that we have an increase in synaptic weight for this typical microzone, or at least for the Purkinje cells, receiving information typically from the other limbs, as well actually as cortical information. Because actually, if you remember in this area, P1 plus, P2 plus, you also have a lot of cortical information from the cerebral cortex. So my view is that to make the adaptation works, the animal had to collect more information from the other limbs. That's why we got these connectivity maps with a lot of granular input from many other areas that were not there before in the control situation, for example. But it's a speculation, I have to say.
Other questions for Philippe?
Yeah, I'll ask a question, Philippe. Hi, it's Sasha.
Hi, Sasha.
Hey, here I'll on whatever view myself. Hey, that was just spectacular. Really beautiful, yeah. Like, that's why I think everybody is so silent. It's like, well, so okay, so internal models. I really like the idea that it's just an adjustable map. And so and you said in your cautious way that one part of the model is stored here in the balance of excitation and inhibition, the dynamic balance. Where else are the internal models stored? Can you expand your view, which has been just so beautifully focused on this little synapse, baby little synapse, and it's like super important. So where else are fundamental aspects of the model? Just to expand your view, or our view. Thank you.
I have to say it's a fantastic question because we can speculate a lot on this.
Of course.
And I would suspect that the whole cerebellum involved in locomotor control or control of limbs is involved into this. I mean, I can expand a little bit. If you remember some data showing that clambin fiber inputs can be observed all over the place in a given movement, meaning that many different microzones actually are active at any time point during the movement. So I would suspect that all these microzones, in a certain way, are organized like a piano, actually in controlling the movement. So if I have to expand our work, I would suspect that if we could record actually in many other lobules and many other microzones in the cerebellum, we would also see adjustment. I would also see some adjustment in the connectivity maps. And I would love to do that, actually, recording many other places, lobule 6 and the paravermis, for example, and lobule 3 and lobule 6, 7, or any other places in cerebellum. I would expect that we could see a little bit adjustment. So I would say, I mean, we know that there is big area, actually, but based on fMRI data now on humans, for example, you have this big area of the cerebellum involved in a particular behavioral task. They cover a huge area. So it's not possible to be so precise that to say that, okay, this is this point here in which the internal model actually is stored for this particular behavior. No, it's probably all over a very different area. But I mean, typically we have to be reductionist. So we have to focus on a single area to try to address, at least the physiology of the system. But I would say imaging data, global imaging data, mesoscale data could be interesting in trying to understand this and try to correlate, function or physiology with behavior in that respect.
Beautiful, thank you.
Shogo has raised his arm. Shogo, please go ahead.
Thank you, Reza. Thank you very much for your beautiful presentation. So I have a question about two things. And one is local connectivity. In your presentation, you showed there is a local connectivity, and local connectivity decreased after the adaptation. So is that the typical change in the after adaptation? If that is true, I think it may be interesting because after adaptation, the connectivity can be wider. And the local connectivity is more decreased after adaptation. So is that the standard typical change after adaptation?
I would not say that local connectivity decreased. I also would like to stress the fact that I showed a lot of z-score, which are not typically the good metric to compare the size of the synaptic rate. So what I observed and what we observed more clearly is that we always see local granule cells connected to the Purkinje cells. So it's a kind of non-conditional input, which makes sense actually in a certain way, because this is in a given microzone, Mossy fibers actually have been demonstrated to connect to a local interneurons and to somehow Purkinje cell as well. So there is a kind of non-conditional communication, and then communication from the other microzone. But what I can say that, yes, I agree with you. After adaptation, you have more microzones connected to the local microzone, because we can maybe speculate and say that it's because we need more coordination, typically in this very invasive situation of the cuff, for example.
I see. Thank you. Yeah, I think that really makes sense. And also another question is about the connectivity map and the Zeppelin, the compartment. So in your cluster in the connectivity map, it seems the cluster border is not corresponding to the Zeppelin map. So instead, between the one Zeppelin positive zone and the one Zeppelin negative zone has very similar connectivity map. It's very interesting, but I think it's surprising that it suggests even between the Zeppelin positive and negative zones, the inputs are very shared, and but the output should be different, right? Because those are the different micro zone. So is that correct understanding? even the zeppelin positive and negative zones share the really the similar input.
Yeah. That's a nice question as well, because in this typical area, Mossy fiber inputs do not match actually with the brain pattern. Right. So, and you have spinocerebellar, external cuneate, cervical as well, and actually also cortical. And they do not perfectly match with the boundaries of the brain, probably because the P1 negative area in that lobule, or in more proximal lobule, you have to subdivide the negative zone into more microzones. So that's I think that this is one explanation of what we observe, that the boundaries do not match with the zebrain boundaries for the Purkinje cell groups having specific connectivity maps. But it's an interesting point, because I think we need to dig more into this anatomy of the fine anatomy of the microzones in that area.
I see. Yeah, thank you very much. That's really great. Thank you.
Any other questions for Philippe? Philippe, thank you so much. I don't hear any other questions. If there are questions, please raise your hand. Otherwise, let me thank Philippe for the brilliant presentation. I so much enjoyed learning about the connectivity maps. And Philippe, thank you so much.
And thanks a lot, Reza, for this invitation and all the series. Actually, there's a lot of information in this YouTube website now.
Excellent. Yeah, and you will be a part of it. It's a pleasure to have you, and I'll see you in August.
Yes, with pleasure. Yes, all my pleasure. Bye-bye.